So we can begin. Today we are going to start a series of lectures in basic tissues. You people have already had a lecture on cell biology. This week and next week we'll be focusing on basic tissues. But just as a matter of introduction, before I focus on the subject matter of today, we know that the body is organized in a particular hierarchy where we have smaller things called atoms, and you people are just from high school, majority of you. So you know examples of atoms. And we know that when atoms combine, they form molecules. Atoms being the smallest unit of an element that participates in chemical reactions. Molecules being the smallest unit of an element or compound that can exist on its own. And again, you know examples of molecules. When molecules combine, they form macromolecules. The macromolecules that build up our bodies are about 4: carbohydrates, lipids, proteins, and nucleic acids. So they are bigger molecules. That's why we call them macromolecules. Remember those 4 macromolecules. When macromolecules combine, they're the ones that constitute organelles. And again, you know examples of organelles. When organelles combine, they form the cell. We have very many cells in the body. Very many, billions of cells in the body. Even though there are such a number of cells in the body, we can classify these cells into various types. And if you are to classify the different types of cells in the body, maybe there will be about 200 plus or minus different varieties of cells in the body. Even though there are about 200 plus or minus different varieties of cells in the body, we can classify these cells or group them into tissues. And we'll get about 20, again, plus or minus, different types of tissues in the body. Although there are about 20 different types of tissues in the body, these tissues can be categorized into 4 basic tissues. Well, for the sake of completion, then, we say that when tissues combine, they form organs. When organs combine, they form organ systems. And we have about 14 organ systems in the body. You people have already been introduced to them, so I'll not go into that again. When the organ systems combine, they form the organism, which is you and me. When we talk about tissues, we're not just referring to cells only. Tissues are described to consist of usually 2 key things, but we add a third one. The 2 key things are the cells and the matrix. The matrix being the material between the cells. So you can use the term extracellular matrix, which means the material outside the cells. Or you can use the term intercellular matrix, which means the material between cells. Is the same thing. So if these are cells, then this region is the matrix. It is outside the cell, or it is between cells, extracellular or intercellular. Don't confuse intercellular with intracellular. Intracellular would mean here. Intercellular means here. Extracellular also means there. Now within this matrix, we may find some different kind of substances, and one of them being water. The water within here is what we are calling the interstitial fluid. That's why some people then talk about tissues consisting of these three: cells, matrix, and interstitial fluid. I've told you that we have about 20 different types of tissues, but these tissues can be grouped into 4 major categories: epithelial tissues, connective tissues, propulsion tissues, and nervous tissue. The objective of today is to focus on the epithelial tissues as well as the propulsion tissues. Next week, you'll focus on the connective tissues. Those ones are those ones are many, so they'll have their own session. There'll be a time that you'll focus on nervous tissue, probably next year. Even though we have 4 basic tissues, these tissues display some unique characteristics in terms of their excitability. Excitability is the ability of a cell to respond to a stimulus by generating an electrical impulse. The excitable tissues, therefore, are tissues which can generate such electrical impulses. In physiology, you'll be told that these electrical impulses are called action potentials. Now, the tissues which can generate electrical impulses are muscle tissue and nervous tissues. Therefore, those ones are called excitable tissues. The tissues which cannot generate electrical impulses are therefore termed non-excitable tissues, and these belong to now epithelial tissues and connective tissues. Right, so I hope that then makes you understand where we are. Today's topic will be on epithelial tissues. This is what you're going to learn in epithelial tissues. We are going to define what epithelium is, and we are going to state the general functions of epithelial tissues. We are also going to talk about the key properties or characteristics of epithelial tissues. We will then explain the concepts which we use to classify epithelial tissues into their various types. After that, then we are going to look at the different types of epithelium. And for each epithelium, we are going to state where they are found in the body. Next, we are going to then look at what we call surface adaptations or surface specializations of epithelium. There is what we call the apical surface, the lateral surface, and the basal surface. And so we look at those adaptations and perhaps their role. Lastly, we'll talk about the organization of glandular epithelium, and we'll be giving examples. So quite a mouthful, but I'll be giving you breaks. So let's start with the first agenda then. We want to define what epithelium is, and we want to know the general functions of epithelium. As a matter of definition, epithelium refers to the layer of cells that cover body surfaces. Like in this case, you're seeing these cells covering body surface. These body surfaces could be an internal surface, like the inside of the intestine, the inside of the heart, the inside of blood vessels, could be those internal surfaces. But they could also be external surfaces, like your skin, the outside of the heart, and the like. So body surfaces are covered by epithelium. Having said so, it then means that wherever you have epithelium, there'll be part of the epithelium that will be free, this side, and there'll be part of epithelium that's attached, this side. The free side is called the free surface or the apical surface. The attached side is called the basal surface of the epithelium. So there is the free or apical surface and the attached or basal surface of epithelium. The part that's attached will be attached to the underlying, most of the time, connective tissue. Having said that epithelium cover body surfaces, I want to ask you a question. And this question, even though it's on your screen, I want to pop something on your screen where you're going to answer so that I can collect the attendance of the class as well. So there's something that has popped on your screen. You have 30 seconds to respond to it. All right, let's proceed. So what are the general functions of epithelial tissues? Now that they cover body surfaces, epithelial tissues will be expected to protect the underlying body tissues. This protection could be from physical injury, could also be from infections. So yes, epithelium has immune functions. Also protects the body from underlying physical injury. Epithelial tissues are also important for absorption of substances from various surfaces. For example, in the intestines, in the urinary passages, and the like. Epithelial surfaces are absorptive. Epithelium also is important in secretion of substances. And this is why epithelium will then form glands which participate in secretion. Some epithelial surfaces are sensory surfaces which detect environmental stimuli. For example, the olfactory epithelium is a sensory receptor that detects the sense of smell. Some epithelial surfaces promote the transport of substances along the surface. This is particularly true for epithelium that is ciliated, epithelium that has cilia. The cilia propel substances to a particular direction, like in the respiratory tree again, the epithelium is ciliated and propel substances along the surface of the epithelium. So these are the key functions of the epithelial tissues. And so I hope if you got the question wrong, you now know the correct answer. We are going to go to the second objective, which is talk about the key properties or characteristics of epithelial tissues. So this is how epithelium would look like typically. The most fundamental property of epithelial tissues is that the cells are closely packed. The cells are very close to each other. This is the most fundamental property of epithelial tissues. And if the cells are closely packed, then it means that the matrix, the material between the cells, is very minimal. And so those two go together as the most fundamental property of epithelial tissues. The third characteristic is that epithelial tissues lack blood vessels. And so because they lack blood vessels, they rely on diffusion of substances. For the epithelial tissues to get nutrients, they'll rely on diffusion of substances, diffusion of nutrients from the underlying connective tissues. Otherwise, you don't have blood vessels in epithelial tissues. Importantly also is that epithelial tissues rest on a basal lamina. This blue thing you're seeing down here is what we call the basal lamina. It's just a thin membrane that separates the epithelium from the underlying connective tissue. This basal lamina is so thin that you cannot see it under light microscopy, but you can appreciate it under electron microscopy. But it separates the epithelium from the underlying connective tissue. We also say that epithelium displays polarity. And what do I mean by polarity? When we talk of polarity, we mean that the epithelium has a free surface and a touch surface. The free surface is what I called the apical surface. The attached surface is what I call the basal surface, as I'd explained to you earlier. Or in this image, you can also appreciate the same. So the free surface is this top part, the apical surface, and the basal surface or the attached surface, this lower part. You can see that black line there is what you're calling the basal lamina. You can call it basement membrane. There's slight difference between basal lamina and the basement membrane. You can check on the difference between the two, but they go together. Last but not least, epithelial tissues display high regenerative potential. It means that the cells have the capacity of cell division, and for that matter, a bit faster compared to other tissues. Well, this is not unique to epithelium. There are other tissues that can also have high regenerative potential, and especially the connective tissues, some of them. All in all, these are the general properties of epithelial tissues. And so we are done with the second objective. Our third objective is to talk about the concepts that we use when you're going to classify epithelial tissues. So this is how we classify epithelial tissues. We can classify epithelium based on the function of the epithelium. And this is the first thing we do. Now, according to this concept, there are two types of epithelium. There's what we call lining epithelium. Excuse me. There's what we call lining epithelium, and there's what we call glandular epithelium. So lining epithelium is typically the one that cover body surfaces. Glandular epithelium is epithelium that forms glands. So that's where we come from. And actually, the subsequent concepts which are used to classify epithelium are largely revolving around the lining epithelium as opposed to the glandular epithelium. And so which are these other concepts? Lining epithelium can further be classified based on two things, mainly, but I'll tell you three. The first one is based on the number of cell layers. How many layers of cells is this epithelium made up of? On this account, if the epithelium is just one layer of cells, then we call it simple epithelium. If the epithelium is more than one layer, we call it stratified epithelium. But there's yet another one, pseudostratified epithelium. I'll be explaining the implications of that. But these are the possibilities. According to the number of cell layers, you can have simple epithelium, you can have stratified epithelium, and you can have pseudostratified epithelium. I'll be explaining this concept shortly. After we've classified epithelium according to the number of cell layers, the second thing you do if you want to classify lining epithelium is to classify it according to the shape of the cells on the free surface, according to the shape of the apical cells. On this account, again, we have three possibilities. Can be squamous shape, can be cuboidal shape, can be columnar shape. Again, I'll be demonstrating to you how these appear. So most of the time, lining epithelium will be classified first based on number of layers of cells, and two, based on the shape of the cells. Most of the time, the story will end there. But there are times where we need to go further beyond these two classifications, because we want to distinguish further between other epithelial types which have similar features in this nature. So on those circumstances, then we have another concept that we use. We want to look at the type of surface adaptation the lining epithelium is displaying. And so some of these will be having cilia, then we say it is ciliated. Some of them may have what we call stereocilia. And I'll be explaining these ones as well at the tail end of the lecture. Some of them have microvilli. Some of them have keratin, so we say they are keratinized. Note that this concept is not used to classify all types of epithelium, but just some few. But this concept and this concept are largely used to classify all types of lining epithelium. So I've told you that we first classify epithelium based on function. And based on function, we have lining epithelium, the epithelium that cover body surfaces, as you can see pointed there. But you also have glandular epithelium, the epithelium that forms glands, as you can see in this image. And that this is the overarching classification of epithelium. But subsequently, if you want to focus on lining epithelium only, then we really don't use these ones then. So we just focus on the lining epithelium. So therefore, the other classifications are based, or the other concepts are based on further classification of the lining epithelium. We can classify lining epithelium based on the number of cell layers, as I told you. If it's made up of one layer of cells like these ones, resting on the basal lamina. The red one is the basal lamina. Then we call this one simple epithelium. If it is made up of multiple layers of cells, as you can see here, the concept here is that it's more than one layer of cells. And when it's more than one layer of cells, you realize that not all the cells will therefore rest on the basal lamina, as in this case. If that be the situation, then we call it stratified epithelium. So in stratified epithelium, you have more than one layer of cells, and that there's no way the cells on the apical side will then rest on the basement membrane or the basal lamina, as opposed to the last one, which we call pseudostratified epithelium. If you look at this arrangement of cells, you can see multiple nuclear layers. And so it gives you the appearance of stratification. But if you look at it keenly, you notice that all the cells are actually resting on the basal lamina. Now that all the cells are resting on the basal lamina, it simply means that it is one layer of cells. So basically, it should be simple epithelium, but it's not similar to this one. You see, in this simple epithelium, the nuclei are in almost one level. But this one, you see multiple levels of the nuclei. In pseudostratified epithelium, you have a mixture of short and tall cells. And for that reason, their nuclear levels are different. So when you look at it under light microscopy, it will give you the impression that it is stratified, but it is not stratified. And that is why we call it pseudostratified epithelium. So based on the number of layers of cells, we have these three possibilities. Based on the shape of the apical cells, we have these three possibilities, as I've told you. When we say epithelium is squamous, it means the cells are flat. Don't confuse with square. The term squamous means flat or smooth. So when the cells are flat, like this, then we call that squamous. Cuboidal would look like this, basically cubes, so the height and the width are almost the same. Columnar would look like this. The height, the cells are taller than they're wide. That's columnar. Now, you know that it is not possible to resolve the cell membrane using light microscopy. And so when you look at this type of epithelium in light microscopy, you may not be able to really appreciate the shapes per se. One of the things that you can rely on is the shape of the nucleus. If a cell is flat, then the nucleus will tend to be horizontal like that one. Not all the time, but a number of times, the nucleus will tend to be horizontal like that. If the cell is cuboidal, then the nucleus will appear to be round or ovoid. When the cell is columnar, then the nucleus will tend to be vertical like this. So that's one of the other concepts we can use to distinguish these shapes of cells. It might not be 100%, but some significant level of confidence. You can use the nuclear shape to depict the shape of the cells. And that will apply for situations where you are not able to really appreciate the shape of the cell on light microscopy because of what I've told you. Light microscope does not resolve the cell membrane. Right, so I've told you the concepts which we use to classify epithelial tissues. I now want us to then look at the different types of epithelium. based on those concepts that we've used. And we are going to state the distribution of each. Now, this objective is largely going to focus on the different types of lining epithelium. So the first one looks like this. So in this case, we have flat cells. As you can see here, one layer of flat cells resting on the basal lamina. So according to the number of cell layers, we'll call this epithelium simple. And according to the shape of the cells on top, we will call this one squamous. So the name of this epithelium is simple squamous epithelium. It simply means that we have one layer of flat cells. Where do we find simple squamous epithelium in the body? So I'll be talking about the distribution now. Simple squamous epithelium is found in the endothelium. The endothelium refers to the inner lining of blood vessels. It is also found in the endocardium. The endocardium is the inner lining of the heart. We find it in the mesothelium. Mesothelium refers to the membrane that lines body cavities. There are three body cavities. Peritoneal cavity, that's the cavity around the intestines. And the membrane there is called peritoneum. So peritoneum is mesothelium. The second body cavity is the pleural cavity. The pleural cavity is the cavity around the lungs. The membrane there is called the pleura. So pleura is mesothelium. The third body cavity is what we call the pericardial cavity. The pericardial cavity is the cavity around the heart, and the membrane there is called pericardium. So pericardium, peritoneum, and the pleura are examples of mesothelium, simple squamous epithelium. The alveoli of the lungs are also made up of simple squamous epithelium, and you understand they'll form a thin barrier for gas exchange. And lastly, we have the Bowman's capsule, also being lined by simple squamous epithelium. And the image we see here is actually the image of the Bowman's capsule. Well, there are other things, but I want you to focus on this one. You see, this is the glomerulus, and this is the Bowman's space. Now look at these cells, the cells that line the space. This is the simple squamous epithelial lining, the one I'm outlining for you. This is simple squamous epithelial lining of the Bowman's capsule. Not these ones. These ones are renal tubules, different epithelium, but this one here specifically is simple squamous epithelial lining. So histologically, simple squamous epithelial lining would look like that. These are just more images to display the same. This is the layer of cells, one layer of cells. As you can see, they are flat. They cover the Bowman's capsule. This is Bowman's capsule. The one on your right is the lung alveoli. You may not appreciate that nicely because of the level of magnification, but you can see this is a thin layer basically. So this is still simple squamous epithelium of the lung alveoli. That is the first type of epithelium we've talked about. So the second type of epithelium we're going to look at is this one, where, look at it, we see the cells here are cuboidal in shape. And we also see that it's one layer of cells. So you just want to call it simple cuboidal epithelium. This is how simple cuboidal epithelium would look like generally. That one layer of cuboidal cells. So this is the lumen, this is the layer. This is the attached side, this is the basal side. Now here you're seeing multiple lumens, that's why you're seeing look like there are two, but no, this is the free side of epithelium, this is the attached side for this epithelium, which is different from this one. Free side, attached side. So where do you find simple cuboidal epithelium? Basically, we find simple cuboidal epithelium in the renal tubules. When you talk of renal tubules, you're referring to the nephron. Actually, these images are images of the nephron of the kidney. Apart from renal tubules, the simple cuboidal epithelium is largely the epithelium that forms the glandular epithelium. Most of the time in the glands, you'll find simple cuboidal epithelium. Simple cuboidal epithelium is also found in ducts of exocrine glands. The ducts of exocrine glands. And lastly, we also find in the thyroid gland. Now you realize that the last three are largely revolving around glands. So to keep it simple for you, simple cuboidal epithelium is in the renal tubules. These are more images of simple cuboidal epithelium. The one forming renal tubules, basically cut in cross section, the first image. On the second image, the one in the thyroid gland, simple cuboidal epithelium. The third type of epithelium I want us to talk about is this one, where we have one layer of tall cells. When you have one layer of tall cells, then you want to call it simple columnar epithelium. This is how simple columnar epithelium would look like. You can appreciate what I told you about the shape of the nuclei when you have columnar epithelium. You can also appreciate that the nuclei tend to be in one level, and that's why we are calling it simple. We find simple columnar epithelium in many places. We find in the female reproductive tract. Which part of the female reproductive tract? We find it in the fallopian tube. We also find it in the inner lining of the uterus. The inner lining of the uterus is called endometrium. So you find it in the endometrium. And lastly, still within the female reproductive tract, we find it in the upper part of the cervix. The upper part of the cervix is known as endocervix. So those are three regions within the female reproductive tract where we find simple columnar epithelium: fallopian tube, endometrium, and endocervix. When we ask you components of the female reproductive tract, don't mention things like ovary. You know, that's not a tract. You need to understand what you're being asked. Second region where we find simple columnar epithelium is the gastrointestinal tract. And so which parts of the gastrointestinal tract really have simple columnar epithelium? This epithelium is present from the stomach all the way to the rectum. Now you can list those components then. So it means that the stomach has simple columnar epithelium. The duodenum has simple columnar epithelium. Jejunum has it. Ileum has it. Cecum has it. appendix has it, the colon has it, and the rectum has it. All those regions between the stomach to the rectum have simple columnar epithelium. Renal tubules also have simple columnar epithelium. I know we'd mentioned that renal tubules have simple cuboidal, and that is still true. There are some parts of the nephron that have simple columnar epithelium. There's some parts of the nephron that have simple cuboidal epithelium. So they all apply. The ducts of exocrine glands also display simple columnar epithelium, in as much as they may also display simple cuboidal epithelium. So there's some ducts that will be lined by simple columnar. There's some ducts that are lined by simple cuboidal. This is the distribution of simple columnar epithelium. So the images here are examples of simple columnar epithelium. One layer of tall cells. This is most likely the intestine, because you can see that one, which we call the villi. Also here, simple columnar epithelium. You can see the cells there, this layer and that layer. There's a lumen there though. So the next type of epithelium we're going to talk about looks like this. Now I want you to look at this type of epithelium and ask yourself whether, according to the number of cell layers, will you call it simple? Would you call it stratified or pseudostratified? And I'm giving you 10 seconds to make up your mind before I proceed. Please don't write on the chat. I'll not be able to follow. So I'll assume that you've concluded it will be stratified. Okay, so look at this. We see many cell layers, and that the cells on top here are not resting on the basal lamina. So therefore, we will call this stratified epithelium, not pseudostratified, as I've seen those who are not listening right on the chat. This is stratified epithelium. How about according to the shape of the cells? Now, according to the shape of the cells, look at them again, and we're looking at the upper image first. When you look at the shape of the cells down here, they're cuboidal. Therefore, you might be tempted to call this stratified cuboidal, but that will be not correct, because we said when you want to classify epithelium based on the shape of the cells, you look at the cells on the apical side. This is the apical side, this is the basal side. You look at the cells near the top and look at the shape of those cells. They are flattened. And so the correct classification of this epithelium is stratified squamous epithelium. We find stratified squamous epithelium in regions where you need a lot of protection against friction. So body surfaces that experience friction, whether that friction is actualized or anticipated, it doesn't matter. But regions of the body that experience friction are expected to have stratified squamous epithelium. Think about those regions, maybe for the next 10 seconds. Just have them in your mind. Again, you don't have to write in the chat. all those regions we are thinking about that are likely to experience friction are expected to have stratified squamous epithelium. So it's an epithelium that's adapted to withstand friction. There are three varieties of stratified squamous epithelium. One of the varieties is the one displayed below, where yes, you have multiple layers of cells, so it is stratified. The cells on top, look at their nuclei, the shape of the nuclei, it appears flat. So stratified squamous. But there's nothing beyond that. I'm looking at this surface, this free surface. There's no apical surface adaptation. I'd mentioned to you that there are times that we love to use this third concept to classify lining epithelium, the concept of apical surface specialization. Stratified squamous epithelium is one of that kind of epithelium where we want to look at the apical surface specialization. So there is this one where we have stratified squamous epithelium, but there is no apical surface specialization. The apical surface specialization that we tend to have in stratified squamous epithelium is keratin. So this one does not have a keratin layer, therefore we call it non-keratinized. So this type of epithelium, the image below, is what we call stratified squamous non-keratinized epithelium. Stratified squamous non-keratinized epithelium is found in many places in the body. You find it in some regions in the mouth, like the lower part of the tongue will have it. The floor of the mouth will have it. Some parts of the cheek will also have it. We also find it lining the esophagus, and also the one that lines the anal canal. You realize that those regions will experience friction in one or another. Also, within the reproductive systems, there are regions with stratified squamous non-keratinized epithelium. The vagina has this type of epithelium. The lower part of the cervix is what we call ectocervix. It will also have this type of epithelium. The distal part of the male urethra also has this type of epithelium. You again realize that those regions experience friction, whether anticipated or actualized. The epithelium of the cornea is also stratified squamous non-keratinized. Cornea is this transparent thing in front of your eye. You know, the way you blink many times in a minute, those frictions need to be withstood by this type of epithelium. The vocal cords, the part used for talking, also experience friction, and so they also have stratified squamous non-keratinized epithelium. So that is one variety of stratified squamous epithelium. And this variety would look like this. You have stratified epithelium. As you can see, the cells on the apical surface appear flat, and this is one of the areas then you rely on the orientation of the nucleus to tell that these apical cells are flattened. Even this is also an example of stratified squamous epithelium. This is specifically that of the esophagus, stratified squamous non-keratinized epithelium. This is also another example, the image on your left, stratified squamous non-keratinized epithelium. You can see in many layers of cells, the apical cells are flattened. The second variety of stratified squamous epithelium is the one that has keratin layer. And look at this. This is the keratin layer. Look at how massive that layer is. How do you know this is keratin? you can't really see nuclei of cells here. So it's just an amorphous material. You see the layers of cells end here. So beyond there, you don't see any cells. You don't see the nuclei. So this is the epithelium from there to somewhere here. That's the epithelium, highly compacted region. The cells on top are flattened. The magnification here is a bit low because I wanted to see the keratin. So you may not be able to appreciate the shape of the cells nicely, but the apical cells are these ones. They appear flattened compared to the basal cells, which are these ones. Then on top of the apical cells, you have this amorphous material, which we are calling keratin. So the name of this epithelium is stratified squamous keratinized epithelium. This epithelium is unique to the epidermis of the skin. So this is how stratified squamous keratinous epithelium will look like. This is the keratin layer. Look at how massive it is. This is the cellular layer. Even this one, this is keratin layer, and this is the cellular layer. This is connective tissue. So we are focusing on the epithelium itself. This is. Okay, third variety of stratified squamous epithelium. We call it stratified squamous parakeratinized epithelium. So in this type of epithelium, we still have stratified squamous epithelium, yes. Then you have a thin layer of keratin. This, I don't know whether to call it red or whatever color you want to call it. This amorphous material here is still keratin. But if you compare the amount of keratin here and the amount that you've seen in the previous slides, you agree with me that this is less, much less compared to what you've seen in the previous slide. When you have this thin layer of keratin, then we call it parakeratinized epithelium. And this type of epithelium is unique to the oral cavity. There are some parts of the oral cavity that are lined by stratified squamous parakeratinized epithelium. The same way, there's some parts of the oral cavity that are lined by stratified squamous non-keratinized epithelium. So really, which parts of the oral cavity will be lined by stratified squamous parakeratinized epithelium? The regions of the oral cavity that are lined by this are regions which come to contact with food. And so this type of epithelium is otherwise known as the masticatory epithelium. Masticatory epithelium is stratified squamous parakeratinized epithelium, the epithelium that comes to contact with food in the mouth when you are chewing. So these are more examples of how stratified squamous parakeratinized epithelium would appear. A very thin layer of keratin, as you can see in these images. Don't confuse this epithelium with stratified squamous keratinized epithelium, where the keratin layer is usually very thick, almost the size of the cellular layer or even bigger. Okay. The next type of epithelium I want to talk about is pseudostratified columnar epithelium. I should have hidden this, then ask you, but okay. Now look at this epithelium. First, let's look at the image on top left. So we see epithelium where you have short cells and tall cells, and that all the cells rest on the basal lamina. So it is pseudostratified. As a concept, if you have pseudostratified epithelium, then which cells will reach the top? It shouldn't be difficult to think. Only the tall cells will reach the top. The shorter ones can't reach the top. So when you have pseudostratified epithelium, you only have one option, columnar. So the image on top shows you pseudostratified columnar. And I'll be telling you how to distinguish pseudostratified columnar from stratified epithelium. Look at the image below. When you look at this epithelium, you notice that we have different levels of nuclei. So it gives you the appearance of stratification. But why are we calling this one pseudostratified as opposed to stratified? Now listen to this, because I don't want to repeat again. When you look at epithelium that has multiple layers of nuclei, in your mind, divide this epithelium into some zones, maybe 4 zones, maybe 3 zones. And this is what I mean. Let's say from here to there, that is first zone, second zone, third zone, fourth zone. That's what I mean. So the zones on the apical side and the zones on the basal side. In stratified epithelium, the nuclei of the cells will be present in all the zones. But in pseudostratified epithelium, the apical zones will be devoid of the nuclei. And that is the only thing you have to distinguish stratified epithelium from pseudostratified epithelium, at least from a light microscopy. The zones which are on the apical side are devoid of the nuclei when you're talking about pseudostratified epithelium. And you can agree with me. Look at this region. We don't see nuclei in that zone. Look at this one. We don't see nuclei in that zone. I know there's that one aberrant one, but largely we don't see nuclei in that zone. This is pseudostratified epithelium. And I've told you, if you have pseudostratified epithelium, it means that you have a mixture of short and tall cells. So if you are to classify based on the shape of the cells that reach the top, it can only be columnar. There are 3 varieties of pseudostratified columnar epithelium. Again, this is another type of epithelium where we rely on the surface adaptation to classify them further, in as much as the other things we also rely on. So now focusing on the epithelium below, the first variety of pseudostratified columnar epithelium is pseudostratified columnar ciliated epithelium with goblet cells. That is the first variety. It means that this epithelium has cilia. As you can see here, this is the cilia. It also means that this epithelium has goblet cells. Goblet cells are mucus cells, like these ones. The ones that don't take the stain, those are the mucus cells. We call them goblet cells. So, pseudostratified columnar ciliated epithelium with goblet cells. This type of epithelium is unique to the airways. So you'll find it in the nasal cavity, you'll find it in the pharynx, you'll find it in the larynx, you'll find it in the trachea, basically the airways. For that reason, because this epithelium is unique to the airways, we call it respiratory epithelium. When we use the term respiratory epithelium, it specifically refers to pseudostratified columnar ciliated epithelium with goblet cells. And this is how respiratory epithelium looks like. You'll be getting this epithelium many times in your exam. You can see these are the goblet cells. It is pseudostratified. Look at the apical zone devoid of nuclei. This is the cilia. So this is pseudostratified columnar ciliated epithelium with goblet cells. Even on this other one, you appreciate the same concept. The second variety of pseudostratified columnar epithelium is pseudostratified columnar ciliated epithelium without goblet cells. This one does not have goblet cells. So first, let's agree it is pseudocertified because we can see the apical zone here is devoid of nuclei. So in as much as you see multiple zones of the nuclei, the apical zone is devoid of nuclei, so this is pseudostratified columnar. You can see cilia there, so it is ciliated, but we don't see goblet cells. Pseudostratified columnar ciliated epithelium without goblet cells is unique to the upper part of the nasal cavity. This upper part of the nasal cavity is the one that functions as the receptor for smell. And so this epithelium is known as the olfactory epithelium. Again, very unique to the upper part of the nasal cavity, the olfactory epithelium. This is how olfactory epithelium will look like in other images, the one on your left, displaying the concept of it being pseudostratified, as you can see. The magnification here does not really allow you to appreciate the brush border, the cilia layer, but you can see nerve bundles here. This is olfactory epithelium. This one shows you the cilia, yes, there. The apical zone is devoid of nuclei, pseudostratified columnar, ciliated epithelium without goblet cells. The third variety of pseudostratified columnar epithelium is the one you're seeing here, where, yes, we are talking about it being pseudostratified columnar, as I've explained to you, but you also see these projections on the surface. These long projections, which appear in groups, are the ones we are calling stereocilia. This is not cilia. Cilia is this one. This is how cilia looks like. Now these ones, they are taller than cilia, and there's some regions which appear to even lack them. They appear like they're in groups. This is stereocilia. Pseudocertified columnar epithelium with stereocilia is unique to the male reproductive tract. When you talk of the male reproductive tract, we are referring to the path followed by semen from the testis to the exterior, but not all the parts of the male reproductive tract have this type of epithelium. So this will be unique to the epithelium in the epididymis, as well as the epithelium in the vas deferens. Epididymis and the vas deferens, those ones have pseudocertified columnar epithelium with stereocilia. Some people want to call this a germinal epithelium. In as much as that could be true, the term germinal epithelium sometimes refer to even the epithelium in the testis, although the epithelium in the testis is not pseudocertified columnar with stereocilia. So use that term germinal epithelium conservatively. This is how the pseudocertified columnar epithelium with stereocilia will look like. First, we agree it is pseudocertified. The apical zone is devoid of nucleus. I can hide that one. Then now I make my case. The same as this one, the apical zone is devoid of nuclei. Yet you have multiple zones of the nucleus, so it is pseudocertified. Then you see projections into the lumen, the apical surface. Those projections are stereocilia. Pseudocertified columnar epithelium with stereocilia. These are histology slides of the epididymis. All right, two more types of epithelium to go. I want to look at the one above. In the one above, maybe I give you a chance to say something. Okay, just a second. Now look at this type of epithelium, the one on top, and ask yourself, is it simple, is it stratified, or is it pseudo-stratified? And your answer should be stratified, because you can see the nuclei are in all the zones, and there are multiple zones. The second question you ask yourself is it stratified cuboidal, stratified squamous, or stratified columnar? And if you look at the cells on top, they appear to have rounded nuclei. I'll go with stratified cuboidal, in as much as some people may go with stratified columnar, and there's no really big deal about that. But importantly, I want to look at the shape of the cells on top, on how the outline of the cells. Basically, you see some bumps, like in that region there, you see a bump. There's a bump there. It's like the cells are popping into the lumen, into the apical zone. When you see those bumpy appearance on the top of the epithelium, it directs you towards a unique type of epithelium that we call the transition epithelium. So how do you pick transition epithelium? It will be kind of stratified cuboidal or stratified columnar. But then, importantly, on top, you'll see a bumpy appearance of the cells, like the top of an umbrella, kind of, or the top of mushroom, those mushrooms that have not yet everted. So that appearance of the top of an umbrella is what gives away transition epithelium. Transition epithelium is called so because there's variation, transition that is, in the shape of the cells, as well as in the number of the cell layers. At some point, this organ will be having, this epithelium will be having like 7 layers, 7 nuclear layers, like maybe in this one, about 7 nuclear layers roughly. But at some point, it could be 3 nuclear layers. So this transition in the number of nuclear layers, 3 to 7. They're all stratified, whether it's 3 or 7. But there's also transition based on the shape of the cells on top. Sometimes they could be columnar, sometimes they could be squamous, sometimes they could be cuboidal. Rarely will you find squamous on a light microscope. When does it become squamous? It becomes squamous when it is stretched. When it is stretched, the apical sides, the cells will be squamous. The reason why you are unlikely to find that in light microscope is because when we prepare the specimens, the stretch goes away, the epithelium contracts, and so it will be either cuboidal or columnar. This type of epithelium is unique to the urinary passages. So you find it in the renal pelvis, ureters, urinary bladder, and urethra. You understand that when the urinary bladder is full of urine, the epithelium will be stretched. At that point, the transition epithelium might have like only 3 nuclear layers, and the apical cells might be stretched flat. When the bladder is not full with urine, the epithelium is contracted. The number of nuclear layers could be 7, like in this case, the apical cells being cuboidal or columnar. Because this epithelium is unique to urinary passages, we call it urothelium. The specific name is urothelium. Transition epithelium is urothelium. The one below shows you a unique type of epithelium where, look, we agree there is stratification. So it's stratified epithelium, but you cannot really pick the specific shape of cells on top. This is the free surface. You can't really pick a particular shape. Because of that, we call it complex stratified epithelium. Complex stratified epithelium is unique to the seminiferous tubules of the testis And so we call it seminiferous epithelium. Right. Let me show the images for that. So this is how transition epithelium would look like. Well, it is stratified, cuboidal, and the cells on top are bumpy, like you can see there, top of an umbrella kind of appearance. This is transition epithelium. This is also transition epithelium. You can see the bumpy appearance of the cells on top. Okay. This is how complex stratified epithelium would appear, specifically the seminiferous tubules of the testis. Now I'm done with that objective, and I can give you a short break. We will now talk about this objective. We are going to look at the surface specializations of epithelium. So before that, it is important that I clarify something. On the surfaces of epithelium. If epithelium is like this, we've said that epithelium rests on basal lamina. This is the basal lamina. Basal lamina is largely secreted by epithelial cells. But there's also another thing called reticular lamina that is secreted by connective tissue. And these two, basal lamina and reticular lamina, is what constitute the basement membrane. So when you say epithelium rests on basal lamina, it's still okay. If you say epithelium rests on basement membrane, it's still the same concept. Now here, when you have this type of epithelium, pick any cell, the top of that cell will be the apical surface of the epithelium. The apical surface, the free surface. And the opposite is the attached part, which are called the basal surface. Because epithelium has an apical surface and a basal surface, we call that polarity. So polarity does not refer to charges, as some of you are asking. It is about the fact that you can define two sides, the apical and the basal side. So this is the basal side. This is the apical side, but there's also this side here, which we call the lateral side. The lateral side is where the individual cells are relating with one another. So when we want to talk about the adaptations, there's some adaptations which are on the apical surface. There's some adaptations which are on the basal surface, and yet there's some adaptations which are on the lateral surface. Let's begin with the apical surface adaptations. Okay, this other image also shows you the concept of the apical surface, the lateral surface, and the basal surface of the epithelium. Let's begin with apical surface adaptations. The adaptations of epithelium on the apical surface are about 4. The first one, the one I'm showing here, is what we call microvilli. Microvilli are finger-like projections of the apical cell membrane. These finger-like projections of the apical cell membrane increase the surface area of the apical cell membrane. And this is an adaptation for absorption. So you'll find it in epithelium that is absorptive. Examples of regions where you'll find this kind of epithelium would therefore be in the small intestines. You'll also find it in the renal tubules. And you'll find it in the ducts of salivary glands. Small intestines, renal tubules, and ducts of salivary glands. the structure of the microvilli is not necessary for you, but you need to know that microvilli are anchored by microfilaments. Microfilaments are actin filaments. They are examples of cytoskeleton. So the cytoskeleton that anchors microvilli are microfilaments, which are also called actin filaments. The second apical adaptation to talk about is this one. This is what we call stereocilia. Stereocilia are just elongated microvilli, very long microvilli, and they tend to exist in groups. We find stereocilia in the germinal epithelium. I told you use the term germinal epithelium conservatively though. So specifically we find stereocilia in the male genital ducts. They're important for absorbing seminal fluid. We also find stereocilia in the ear. And within the ear, they function as mechanoreceptors, the receptors for balance. So don't forget, in the male genital tract for absorption, in the ear as mechanoreceptors for balance. The third apical surface adaptation is cilia. Cilia refer to filamentous projections on the apical cell membrane. Usually, cilia is different from microvilli in two ways. Cilia is motile, which means it can beat in a particular rhythm. Microvilli is not motile. The second difference between cilia and microvilli is that cilia is made up of microtubules. The cytoskeleton that anchors cilia is microtubules. The cytoskeleton that anchors microvilli is microfilaments. Two differences I've told you. We find cilia in regions where we need substances to be propelled. And this is particularly unique in the airways as well as in the fallopian tube. I therefore want you to think through if a patient has a disorder in motility of cilia, and there are such disorders, one of them is called the Cartagena syndrome. How do you expect those patients to present clinically in a patient who has disorder of cilia motility? How do you expect such patient to present clinically? The condition is called Cartagena syndrome. The fourth surface adaptation is keratin. And so epithelium with keratin is described as being keratinized. It simply means a thick layer of keratin. This is an adaptation against frictional forces, and you find keratinization in the skin, wherever it is. We also find keratinization in some parts of the oral cavity regions, which we are calling the masticatory epithelium. That's where you find keratinization. Keratin is an intermediate filament that is found in epithelium. So those are apical surface adaptations. How about lateral surface adaptations? And this is the lateral surface. So in the lateral surface, we have junctions, which we just call intercellular junctions because they are between cells. The intercellular junctions in epithelium are many, but I'll talk about three. This is what we call tight junctions. You can also call them occluding junctions. In Latin, we call them zonular occludens. The tight junctions are regions between cells, like here, that prevent paracellular transport. Paracellular transport is transport of substances between cells. These junctions occlude the space between cells so that substances which need to be transported cannot be transported between the cells. Those substances can only be transported through the cells. If you have sodium that need to move this way, it has to go through the cell, not between cells. Tight junctions prevent paracellular transport. They only allow transcellular transport. The second intercellular junction talk about is adhering junctions, which we call desmosomes. Desmosomes are adhering junctions. They are responsible for holding adjacent cells together. So they anchor adjacent cells together. Those are desmosomes. Usually, each cell will provide something like a disc. So one disc on one side, the other disc on one side, or let's say half a disc on this side, half a disc on this side. The two combine to form desmosomes for holding adjacent cells together. And this is why the epithelial tissue, then the cells are highly compacted because they have adhering junctions, which we call desmosomes. Then we have communicating junctions. Communicating junctions are gap junctions that allow free ionic movement from one cell to another. So these are the lateral adaptations in epithelium. The others, I don't want to talk about them. I've picked these three which you are likely to come across. How about basal surface adaptations? The basal surface adaptations talk about are two. On the basal side of the epithelium, you may have foldings of the cell membrane, so we just call them basal membrane infoldings. You can call it basal membrane invaginations. They increase the surface area of the basal membrane, and that is important again for transport of substances, increasing the surface area for transport of substances. Apart from basal membrane invaginations, the other basal surface adaptation is what we call hemidesmosomes. Now look, for a desmosome, you have a disc, half a disc on one side, half a disc on the other side. They combine to form desmosome for holding the adjacent cells together. Because on the basal side, the epithelium, the cell is now not relating to another cell, but now to the basal lamina, we'll only have half a disc, and that's why we call it hemidesmosome. So hemidesmosome anchors the cell to the basal lamina. A desmosome anchors the cell to the other cell. So hemidesmosome is still an adhering junction. Right. Those are the adaptations of the different surface of epithelium. I want us to now talk about glandular epithelium. When you talk of glandular epithelium, we're referring to epithelium that forms glands. Before we talk about glandular epithelium, it will be important to clarify two concepts. We have different types of glands. There's what you call the exocrine gland, and there's what you call the endocrine glands. The exocrine glands secrete body fluids. The endocrine glands secrete hormones. The exocrine glands release their secretions, these body fluids, into ducts. And these fluids are then transported by these ducts to epithelial surfaces. On the other hand, endocrine glands secrete hormones into the bloodstream. and these hormones are then transported to various target organs. For these reasons, therefore, an exocrine gland have an elaborate ductal system to transport the secretions. On the other hand, an endocrine gland will not have ducts, but instead an endocrine gland will be highly vascularized compared to an exocrine gland. They're still vascularized, but not as vascularized as the endocrine gland. And so I'm giving you 30 seconds to then name examples of each. Just name on your end. You don't have to write on the chat. 30 seconds. Okay, we can proceed. So based on what you've answered, I want to ask you another question. Make sure you respond, because again, it will be part of your attendance. Let me disable the chat so that you don't cheat. So that's a question, but I want you to answer it in the poll. I've launched the poll now. You have 30 seconds. We can proceed. So the images here show you glandular epithelium, how glandular epithelium develops embryologically. You've not done this, so maybe let me not say much. But to distinguish glandular epithelium, the one that form exocrine glands will have ducts, as you can see here. The one that form endocrine glands will have blood vessels around them, many of them, as you can see in these ones. What's the structure of an exocrine gland? I'll be talking more about exocrine glands because endocrine glands will have a lecture for them another time, maybe much later, second semester. But exocrine glands, this is the final thing. An exocrine gland is described to have two components. This section here is the secretory portion. The secretory portion is specifically the one we are calling the glandular epithelium. But we also have a ductal portion. The ductal portion being the part that transports the secretions from the secretory portion to an epithelial surface, as you can see. I mentioned to you that glandular epithelium will tend to be simple cuboidal, and now you understand. Okay, this could be either cuboidal or columnar. They both apply. So the point I want you to understand is that an exocrine gland has two components: the secretory portion and the ductal portion, or the glandular portion and the ductal portion. We classify exocrine glands based on a number of things, and I'll be teaching you 4 concepts which we use to classify exocrine glands. The first concept is based on the structure of the secretory portion. Remember that the secretory portion is kind of the end of the gland, so some people call the end piece. If the secretory portion looks like this, then we call it tubular. In cross-section, a tubular end piece will look like this. This is how it will appear on light microscopy. You can also have an end piece which looks like this, the secretory portion being made up of large cells with the nuclear being mostly on the basal side. If this is the organization, then we call it acinar. Different from tubular. This is acinar. If you look at an acinus in cross-section, it will appear like this on light microscopy. The main difference between this and this is that here, the cells are on the basal side. The lumen is very narrow. The third type of secretory portion is the alveolar type. And alveolar look like this. The difference between this and this is that there's ballooning of the cavity of the secretory portion. And this is how alveoli will look like on cross-section. So we can classify exocrine glands based on the structure of the secretory portion. The second way we can classify exocrine glands is based on the structure of the ductal portion. And here is simple. If the duct is branched, then we call it simple gland. Sorry, we call it compound gland. If the duct is not branched, then we call it simple gland. So based on the branching of the ductal portion, we have simple exocrine glands as the ones on top there. And you have compound exocrine glands like the one below. This is based on branching of the ductal portion. Is the ductal portion branching? It's compound. Is it not branching? Simple. The third concept we use to classify exocrine glands is based on the type of secretion that they give. Now, some of you asked this, and I want to clarify then even here. When you look at these three images, you'll notice that they are made up of mainly acini, the acinus. You remember, I've just told you how acinus look like in cross-section. So these are acini, but they are acini of different types. Some acini are white, which means they're not taking the stain. So that means that they're mucus acini. But there are some acini which are deeply staining, which means they pick the stain. So that means that their secretions don't really have a lot of mucus. So we can classify exocrine glands based on the type of secretion they have. If a gland is predominantly staining, like this one, it means that the secretions is largely watery, and so we call it a serous gland. So these are serous acini. Well, there are a few mucus acini there, but most of them are serous acini. This is the ductal portion, so ignore this one. Now compare that with this one. Now the ones which are staining are ductal portions, so again, ignore them. But look at the acini. Most of them are not taking the stain because they contain mucus. So these are mucus acini. And this is also how the goblet cells will look like within the epithelium, for those who are asking. Look at the third one. The third one has a mixture of mucus and serous acini. So you can call it a mixed gland, or you can call it a seromucus gland. So based on the type of secretion, we have those three varieties of exocrine glands. Lastly, we can classify exocrine glands based on the mode of their secretion. According to this classification, there are three varieties of exocrine glands. The first one is what we call a merocrine gland. What does a merocrine gland do? A merocrine gland releases its secretions without losing any part of the cell. So it means that the secretions are within vesicles, and those vesicles are released into the extracellular space, into the lumen, that is. The cell remains intact. That's merocrine gland. Examples of merocrine glands include most of the sweat glands. As opposed to this second type, which we call holocrine gland. In holocrine gland, the whole cell disintegrates. So the whole cell disintegrates and you have to form new cells. So in holocrine secretions, you have a lot of mitosis taking place to replace the cells which have been disintegrated. Examples of holocrine glands include sebaceous glands. The third type of exocrine glands based on the mode of secretion is what we call apocrine gland. In apocrine gland, a part of the cell is pinched off. Not the whole cell, but a part of the cell is pinched off. I will leave it to you to then find examples of apocrine glands. At least I've given you examples of the first two. So we can classify exocrine glands based on the structure of the secretory portion, based on the branching of the ductal portion, based on the type of secretion, and based on the mode of secretion. So look at various examples that we have here. This one here, this one here is a tubular gland, as you can see. The secretory portion is tubular. So it's a tubular gland. The ductal portion is only one, it's not branching, so it's simple. That's why we call it simple tubular gland. And later on, you can look at those examples when you get the PowerPoints and when you get the recording. Look at the second one. It is still a simple tubular gland, but it is coiling. So we just call it simple tubular coiled. Look at the third one. The ductal portion is one, so that's why we call it simple. But the secretory portion has multiple parts, so it has branched tubular portions. The secretory portion in this case are tubular, but they are branched tubular. We still call it simple because the ductal portion is one. Compare that with this one. It is simple because the ductal portion is one, but look at the secretory portion. It is not tubular in this case, but it's acinar. So this is simple acinar gland. Compare that with the second one, the next one there. The ductal portion is one, but the acini, the secretory portion, are multiple. That's why you give it that name. Now distinguish that with this one, where the ductal portion is branching. You can see this is a duct, this is another duct. So the ductal portion is branching. So these are compound gland. The secretory portion being tubular. So this is compound tubular gland. If you compare that with this one, the secretory portion are acini, kind of, as you can see here. The ductal portion is branching, so this is compound acina. And the last one here, the branching of the ductal portion makes it compound. The morphology of the secretory portion make it be called tubuloacina. Why? Because we see acini, but we also see tubular secretory portion. So this is a compound tubuloacina. Those are the different types of exocrine glands. At your own time, you'll go through this column of examples and the specific features for each. Good. These are other examples of the same concept that I've just told you. We have now finished on organization of epithelial tissues. I have 2 questions for you, I think, on epithelial tissues. Again, you'll answer them in the poll. Let me launch that. There are 2 questions for you, but they're simple, so it'll take 1.5 minutes. You have them on your end right now, on your screen. Take 1.5 minutes. So the first question asked you where we find the simple columnar ciliated epithelium. Now, these are how we ask you the questions. Majority of you have gotten it right, but as you can see, there are still few of you who have failed. On the second question, which is asking you description of epithelial tissues, again, majority of you have gotten it right, but as you can see again, there's some of you who have failed it, and so you need to revisit what you've said in the lecture. All right. I'll give you another break. It's now 10:23. So we come back at 10:35.